You know that feeling when you're trying to make sense of the whole landscape of neuropsychiatric disorders?
Mm-hmm.
Things like schizophrenia, Alzheimer's, autism. It can feel overwhelming, like trying to piece together a puzzle with, I don't know, way too many strangely shaped pieces.
Absolutely. Yeah, it's complex, and for so long, our understanding, it's really been built on observing outward signs, symptoms.
Right.
But imagine if we could zoom way in, like beyond what we can easily see, right down to the fundamental activity within our brain cells. That's kind of the shift we're exploring today.
Exactly. So
Hmm.
let's forget the standard diagnostic boxes for a moment, because
Yeah.
we're diving into a really compelling kind of new way of looking at these complex conditions.
Okay.
Our mission today is to really get our heads around this idea of transcriptomic pseudo-immaturity.
Mhm.
It's a bit of a mouthful, but the core idea is that these seemingly very different disorders might actually share a surprising biological thread.
And that thread seems to be linked to how our brains develop.
Right.
Precisely. We'll be looking at research that asks this really fascinating question. What happens to gene activity in the brain when it's experiencing intense neural activity? Think of it as like a state of being overexcited.
Okay, overexcited.
Yeah. And how does that compare to the normal gradual changes in gene activity that happen as a brain matures, from infancy to adulthood?
So to dig into this, we've got some really interesting studies lined up. We're looking at how genes behave in rat models experiencing seizures, which is a pretty clear example of this neural overactivity, and then comparing those patterns to the gene activity of the developing brains of young mice and even human fetuses.
And what's truly intriguing, what pulls this all together, is this possibility. Could this state, the brain being in overdrive, could it actually push mature brain cells back towards a more immature state, at least at a molecular level?
Wow, okay.
That's what researchers are calling transcriptomic pseudo-immaturity. So our goal in this deep dive is really to see if the shared pattern could offer a more unified way of maybe understanding these diverse neuropsychiatric conditions.
Okay, that sets the stage perfectly. So how did researchers even make that initial leap? I mean, the connecting an overactive adult brain with the processes of a young developing brain. That seems like quite a jump.
It does, yeah. Well, the starting point was to actually create that state of neural hyperexcitation in adult rats.
How did they do that?
They used a compound called pilocarpine. It induces seizure activity, so you get that hyperexcitation.
Got it.
Then the crucial next step was to compare the changes in gene expression, which genes got turned up, which got turned down in these rats, with the gene expression patterns we see during normal brain development.
Okay.
They focused specifically on the dentate gyrus, which is a key area in the hippocampus, really important for learning and memory. And they looked at infant versus adult mice, and also compared the hippocampus in early versus later stages of human fetal development.
Okay. So here's where it gets really interesting for me. What did they actually find when they compared these two situations, the overexcited adult rat brain and the normally developing young brain?
The overlap was, well, pretty remarkable, actually. Quite significant.
Really?
Yeah. A substantial number of genes showed similar directional changes in their activity. So if a gene's activity went up during development, it also tended to go up in the overexcited state, and vice versa.
Huh.
To organize this, they grouped these overlapping genes. They came up with these terms, HIM genes. That stands for hyperexcitation-induced maturity-related genes.
Okay, HIM, maturity related.
Right. These are genes that typically become more active as the brain matures, but what they found was that these genes decreased in activity in both the hyperexcited rats and in the immature brains.
Whoa, okay, so genes that are usually a sign of a more mature developed brain were actually less active in both the seizure model and in the normally developing young brain.
Exactly.
That's counterintuitive. What about the other set?
The other group they called high genes, hyperexcitation-induced immaturity-related genes.
High. Immaturity. Makes sense.
These are genes that are generally more active in the developing brain, and their activity usually goes down as the brain matures. But they found that these high genes actually increased in activity in both the overexcited adult rats and in the immature brains.
That is a really striking parallel. It really does suggest that when an adult brain gets, pushed into this overactive state, it can, at least at the level of gene activity, start to resemble a brain that's still developing.
Exactly right. And this initial discovery, this parallel, was really the springboard for looking deeper. Like, what are these HIM and high genes actually doing biologically? Which is, the next logical step.
Right. So if these HIM and high genes are the key players in this pseudo-immaturity thing, what kind of biological jobs do they typically have? What are they responsible for in the brain?
Yeah, good question. So when the researchers dug into the functional categories of these gene groups, some pretty clear distinctions emerged. Let's start with mice. The Hymin genes, remember, those are the ones that go down in both hyperexcitation and immaturity. They were heavily involved in synapse and channel activity.
Oh, okay. So communication, basically.
Exactly. Think of these as the fundamental communication systems. Synapses are where neurons connect, exchange signals like the handshake between brain cells, and channels regulate the electrical signals flowing through them.
That makes a lot of intuitive sense. I mean, as the brain matures, you'd expect those communication pathways to become more refined, more efficient.
Precisely.
So a decrease in the activity of those genes, both an immature brain and an overexcited one, could indicate maybe some kind of disruption or just a different state of that communication system.
That's a good way to put it.
What about the human Him genes? Did they show a similar pattern? Was it also about synapses and channels?
Yes, incredibly similar. The human Him genes also showed this strong connection to synapse and channel activity. Things like the transmission of nerve impulses, the whole process of synaptic transmission itself, the structure of axons. Those are the long arms of neurons sending signals.
Like the wiring.
The wiring, yeah, and the synapses themselves. This consistency across species, mice and humans, it really strengthens the idea that these specific synaptic functions are particularly maybe vulnerable or susceptible to being pushed towards a more immature-like state by this neural overactivity.
Okay, so Him genes are centered around how brain cells talk to each other. Synaptic communication.
Yeah.
What about the high genes, the ones that increase in both conditions? What roles do they play?
Right, the high I genes. In both mice and humans, these were strongly associated with the nucleus.
The cell's control center.
Exactly, where the DNA is housed. In mice, they were linked to processes like the cell cycle and DNA replication, things that are really active during brain development when cells are rapidly dividing and growing.
Okay, that makes sense for development. And in humans, were the high genes doing similar things there?
In humans, this connection to the nucleus was, if anything, even stronger. The human high genes were heavily associated with the cell cycle, with chromosomes, the structures carrying our genetic blueprint, and even with the cell's response to DNA damage. Again, these are processes that are absolutely crucial during development, but generally much less active, kind of quieted down in a mature, stable brain.
So we've got this interesting split. One set of genes, the Him ones, related to brain communication, becomes less active in both immaturity and overexcitation. And another set, the high I ones, tied to fundamental stuff in the nucleus, becomes more active. That functional difference seems really significant.
It really is. It suggests that this overexcitation isn't just like a blanket effect on all gene expression. It seems to be selectively impacting different biological processes. It's potentially nudging the brain towards an immature functional state in some areas, like communication, while maybe activating these developmental-like processes in others within the nucleus.
Fascinating. And the study also looked at the timing and location of these changes, right? That adds another layer.
Yes, exactly. They observed that in the mouse model, the changes in Him gene activity, those synaptic ones, they seem to appear earlier after the seizure, and they stabilize more quickly compared to the changes in the high I genes.
So the communication-related changes happen faster.
Appeared to, yes.
Oh.
And spatially, they also found differences. The protein products of Hymin genes tended to be more present at the plasma membrane, the cell's outer layer, where a lot of the synaptic machinery actually sits.
Right, where the action happens.
Yeah, whereas the high I gene products were more often found inside the nucleus. This just further emphasizes the distinct roles and kind of dynamics of these two sets of genes.
Okay, so we're building this picture now. Neural overexcitation seems to cause this specific shift in gene expression in adult brains, making them resemble immature brains in key ways, affecting both how cells communicate and these fundamental processes inside the cell nucleus. Now, for the really big question, how does this idea connect to actual human neuropsychiatric disorders?
Right. This is where it gets really clinically relevant, potentially. The researchers introduced what they called the Him index and the high I index.
Okay, indexes, like scores.
Exactly. Think of them as scores. They tell us how much the gene expression patterns seen in a specific disease overlap with those Him or high I gene sets we've just been talking about.
Okay, so how does that work?
Well, a high Him index in a disorder would suggest that genes that normally become more active as the brain matures are found to be less active in that particular disorder. similar to what we saw during overexcitation.
Okay, so lower maturity markers.
Right. And conversely, a high I index would suggest that genes that normally become less active with maturity are found to be more active in the disorder. Again, mirroring that overexcitation effect and the immature state.
Gotcha. And to visualize this, they used some kind of 2D analysis, plotting these disorders based on their HIM and high index values.
That's right, a two-dimensional plot. And when they did this for different neuropsychiatric conditions, some interesting patterns started to emerge.
Let's hear about them. What about, say, schizophrenia?
For schizophrenia, the most consistent finding, looking across multiple different data sets, was a dominant HIM index.
High HIM index, so lower activity in those maturity-related genes.
Exactly. Think of it like this. The genes that should be the hallmarks of a fully developed brain in terms of their increased activity seem to be underperforming or less active in schizophrenia, almost as if the brain hasn't quite completed a key stage of its molecular maturation in those specific areas.
Right.
And crucially, this same pattern of underperformance is what they saw triggered by overexcitation.
That's a really interesting connection. So in schizophrenia, it's almost like the brain is stuck or reversed in terms of expressing these specific maturity genes, and that's a pattern linked to overactivity.
Yep.
Okay. What about Alzheimer's disease?
Alzheimer's showed a more complex picture, actually.
Oh.
Brain samples taken directly from Alzheimer's patients, they tended to have a high HIM index, kind of similar to schizophrenia.
Okay.
However, when they looked at neurons that were grown in the lab from induced pluripotent stem cells, iPSCs, derived from Alzheimer's patients, those cells showed a high I index instead.
Oh, that's different. High means more activity in the immaturity-related genes. Why the difference?
Well, it's hard to say for sure. It might reflect the different cell types being studied whole brain tissue versus specific neurons grown in a dish. Or maybe it relates to different stages of the disease progression. It really highlights the complexity, of studying these conditions.
Yeah, it definitely underscores that we're looking at intricate systems. Things aren't always straightforward. What about ALS, amyotrophic lateral sclerosis?
In ALS, the data sets they analyzed often showed a dominant high I index.
High, so the opposite of the main schizophrenia finding.
In a way, yes. It suggests that in ALS, many of the genes that normally decrease with maturity are found at higher levels. And this increase is also seen in the context of neural overexcitation. So yeah, almost the opposite molecular signature to what they generally observed in schizophrenia, at least in terms of this HIM-high balance.
Wow. Okay, so almost a flip side to schizophrenia at this molecular level. What about autism spectrum disorder? That's another condition with a lot of ongoing research.
Right. The initial analysis for autism, looking across various data sets, didn't reveal a really clear dominant pattern. It was a bit mixed. However, the researchers later focused on a more tightly controlled data set, one where they had maybe better consistency in samples and methods. And in that data set, they did find a high index in autism.
Okay, so maybe leaning towards that immaturity signature like ALS, but perhaps more subtle or context dependent.
Possibly. It suggests that while the initial picture was mixed, there might still be a link to this immaturity-related gene expression pattern in autism as well, particularly when you look at certain types of data or perhaps specific subtypes.
Interesting. And what about some of the other major neuropsychiatric disorders we hear about, like Parkinson's, bipolar disorder, Huntington's, major depressive disorder? Do they fit into this HIM-I framework?
Generally, these disorders showed less pronounced HIM or high index values in this particular analysis. They didn't cluster as strongly towards one end or the other.
So a weaker link.
It suggests that this specific form of pseudo-immaturity, the kind that seems inducible by neural overexcitation, might have a weaker connection to these particular conditions compared to, say, schizophrenia, Alzheimer's, ALS, and potentially autism. Doesn't mean there aren't other molecular changes, just maybe not this specific pattern.
It's fascinating, though, seeing these different disorders potentially clustering along these molecular axes of like maturity and immaturity in their gene expression. And you mentioned they also looked at a more controlled data set that kind of backed up some of these initial findings.
Yes, exactly. They analyzed a well-regarded data set from Gandel and colleagues. This was a study where significant efforts were made to account for potential confounding factors, things like medication effects or post-mortem interval.
And what did that show?
Well, this analysis supported the HIM-dominant pattern in schizophrenia and the high I-dominant pattern in autism, which fits with the other findings. But interestingly, it also revealed a HIM-dominant pattern in alcohol abuse disorder.
Oh, interesting.
And a high I-dominant pattern in inflammatory bowel disease.
Wait, inflammatory bowel disease, that's not even a brain disorder.
Right. It suggests this concept, this pattern of pseudo-immaturity linked to him and higher genes, might actually extend beyond just the traditional neuropsychiatric categories. Maybe it reflects some more fundamental biological stress response.
That really broadens the scope.
Oh.
Okay, this is painting a really compelling picture. But how much of this evidence comes from studying humans directly versus using animal models? Did they see similar patterns in animal studies designed to mimic these disorders?
Absolutely, that was a key part of validating the idea. They looked at various animal models. For instance, in models of acute brain injury, things like seizure, stroke, which they call ischemia, and infection, they observed a change in the HIM and high indexes over time.
A change over time. How so?
Well, often, right after the injury, they'd see a more HIM-dominant pattern. But as time wore on, maybe days later, the pattern often shifted towards being more high-dominant.
So the response evolves.
Exactly. That temporal dynamic is quite interesting. It suggests the brain's molecular response to these acute stresses isn't static. It changes, and maybe that shift reflects different stages of damage or repair processes involving these maturity-immaturity gene sets.
That time-dependent shift is intriguing. It suggests the brain's molecular response to acute stress might evolve in terms of this maturity-immaturity signature. What about the genetic animal models, the ones specifically designed to mimic certain neuropsychiatric disorders by carrying specific gene mutations?
Yeah, they examined several of those too. In a mouse model of ALS, one that carries a specific mutation in the SOD1 gene known to cause ALS in humans, they consistently found a dominant high I index.
Okay, high matching the human ALS data.
Exactly, which strengthens the connection. In mouse models of Alzheimer's, ones with mutations in the APP and PSCN1 genes, they saw a dominant high index in the cortex region of the brain. The hippocampus, though, showed a more mixed pattern.
Which again sort of mirrors the complexity seen in the human Alzheimer's data with the brain samples versus the iPSC neurons.
Precisely. It shows that even within the same model, different brain regions might respond slightly differently.
Makes sense. And what do they find in the animal models relevant to schizophrenia?
So in mouse models with genetic alterations linked to schizophrenia risk in humans, like the DF16A heterozygous mice and the ICAM-KII plus mice, these showed a dominant HIM index.
High HIM, consistent with the human schizophrenia data again.
Yep. Very consistent. There was another interesting model, the Shenatu knockout mouse. These mice exhibit features of both schizophrenia and intellectual disability, and they showed high levels in both the HIM and high indexes.
Oh, high in both, so a more complex disruption.
Exactly. It suggests maybe a more widespread or different kind of dysregulation in that specific model, impacting both sets of genes strongly.
It sounds like there's a pretty good level of agreement between some of these key animal model findings and the human data. That definitely seems to strengthen the argument that this transcriptomic pseudo-immaturity might be a real underlying mechanism in these disorders, not just a correlation.
Oh, it really does. And the fact that they observed these patterns in animal models, where researchers have much more control over factors like medication history, diet, environment, it adds considerable weight to the idea that the disease mechanisms themselves are intrinsically linked to this pseudo-immaturity phenomenon. It's less likely to be just an artifact of treatment or other human-specific factors.
Right. It points more towards the core biology of the disease.
Yeah.
Okay, so after pulling all this together, the human data, the animal models, the functional analysis of the genes, what are the key takeaways here? What are the potential implications of thinking about neuropsychiatric disorders through this lens of transcriptomic pseudo-immaturity?
Well, I think the main takeaway is this really intriguing possibility that this specific pattern of gene expression, one that can apparently be triggered by neural overactivity and which resembles a more immature brain state at the molecular level, seems to be a shared feature across several seemingly distinct neuropsychiatric disorders.
Like schizophrenia, ALS, maybe Alzheimer's and autism.
Exactly. And this suggests that despite the really wide range of clinical symptoms we see in these conditions, there might be some common underlying biological pathways at play related to this maturity-immaturity balance.
That could be a real game changer, couldn't it, for how we approach these conditions? I mean, instead of just grouping them strictly by symptoms, which can overlap and vary so much,
Right.
we might be able to identify more biologically coherent subgroups based on these shared molecular features.
Yeah.
Like maybe there's an HIM-dominant type of illness and a high-dominant type cutting across current diagnoses.
Exactly. That's the potential implication. This could lead to a more biologically informed way of classifying these disorders, maybe moving us beyond our current reliance on purely symptom-based diagnosis, which we know has its limitations.
Yeah.
And it also just opens up new avenues for understanding the fundamental biological basis of these conditions. What goes wrong at this very basic level of gene regulation?
But of course, with any new and exciting area of research like this, there are always limitations to keep in mind. What did the study authors themselves point out as, potential caveats or areas needing more investigation?
They were quite upfront about several limitations, which is good science. First, the number of available data sets for some disorders is still relatively small. So more data, especially well-characterized data, is always beneficial to confirm these patterns.
Sure. More data is always good.
Then there's the inherent variability in the types of samples used across different studies. Some looked at whole brain tissue homogenates, others focused on specific brain regions like the cortex or hippocampus, and still others used neurons grown in a lab from stem cells. These aren't all the same thing.
Right, comparing apples and oranges sometimes.
A little bit, yeah. And in the human studies, there's always the challenge of potential confounding factors, like the impact of long-term medications on gene expression. It's hard to disentangle disease effects from treatment effects.
Always a tricky one in human brain research.
It is. And finally, while comparing gene expression across different species like rats, mice, and humans is incredibly informative, you always have to remember there are biological differences between species. A gene might not have the exact same function or regulation across the board.
Those all sound like fair points. Important things to keep in mind as the research progresses. However, they did point out a particular strength in their methodology, didn't they? Something about the statistics.
Yes, they emphasized the strength of the statistical method they used, called the Running-Fisher test.
Running-Fisher test, okay.
Its value is that it allows for meaningful comparisons across different data sets, even data sets generated using slightly different technologies or coming from different species. It's designed to look for consistent enrichment patterns, which is crucial when you're trying to synthesize findings from a variety of studies like they did here. It helps find the signal through the noise.
Okay, that makes sense. It helps bridge those gaps between different studies and species.
Right.
So to sort of bring it all together then, this deep dive into transcriptomic pseudo-immaturity has uncovered this really thought-provoking idea that some aspects of major neuropsychiatric disorders might involve a kind of, rewinding or maybe a stalling of brain maturation, right down at the level of gene expression.
Yeah, molecular echo of development, perhaps gone awry.
And that neural overactivity, that hyperexcitation, could be a factor that either triggers this or maybe makes it worse.
Precisely. It really does open up a potentially new way of thinking about the underlying biology of these incredibly complex conditions.
Absolutely. And it leaves us, and you listening, with a really interesting question to ponder. Could understanding this immature genetic signature, this shared pattern that seems to cut across different traditional diagnoses, could that ultimately pave the way for new diagnostic tools?
Or even new therapeutic approaches.
Right. Therapies that maybe target these fundamental biological mechanisms as pseudo-immaturity, rather than just trying to manage the outward symptoms we currently focus on. Could we maybe encourage the brain to remature its gene expression?
That's the long-term hope, I suppose. It definitely feels like this deep dive has only scratched the surface of a much larger and potentially transformative area of research into the brain, its development, and what happens when things go off track in neuropsychiatric disorders.
A fascinating layer of complexity indeed. Lots to think about.